glucose, glucose is like very, very common. Almost every living thing uses glucose as a source of energy, including ourselves. Now, glucose comes in two isomers or comes in two versions. There's this version you find like in food, you find in starch and rice, and you have another version of it, which is like this. Do you recognize, can you spot the only difference between this glucose on the left? I'm going to call that one on the left, I'm going to call it alpha, and beta. Where's the only difference I have between alpha glucose on the left and beta glucose on the right? Very good, Sama. It's the first carbon. So carbon number one makes a big difference. Here, the OH is pointing down. Here, the OH is pointing up. All right? So alpha glucose has OH down, beta glucose has OH up. Now, where's that beta glucose found? Beta glucose is rare. So that's like a rare case we have here. It's only found in plants. Particularly, it's found in the cell wall of plants. Okay? We don't deal much with beta glucose. We're going to keep beta glucose until unit 2. But for now, just learn how you would identify them. The only difference you have between alpha and beta is this first carbon. If it's alpha, the OH is down. If beta, the OH is pointing up. Let me show you why sugars are soluble, right? We've seen why salts dissolve, but why sugars are also soluble in water. Remember the line we talked about here when we were doing electrophoresis? What did we say? We say, for a molecule to dissolve, it must bind to water, right? So that's the case here. The case we have here is that glucose dissolves in water. The reason why glucose dissolves in water is because the water attracts the water molecule. The sugar attracts the water molecules. Oxygen to hydrogen, oxygen to hydrogen. Do you recognize what's going to happen now? You're going to form hydrogen bonds here. So we're going to end up with hydrogen bonds between sugar and the water. So the reason someone asks you, why do sugars dissolve in water? Simply tell them, sugars have plenty and plenty of OHs, and those OHs form hydrogen bonds with water. I want to try something. I want to do the condensation reaction, just like the one we've done earlier. I want to do this between two sugars. So I have like alpha glucose on the left, and I have another alpha glucose on the right, and I'm going to join them up together. I'm going to form like a hydrogen bond. Sorry, I'm going to form a bond between those two, a real bond, not a hydrogen bond, okay? How do you do this? Again, I'm going to link up this carbon and this carbon. What carbon do I have on the right? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, and 6. Right? So it's going to be linking up carbon 1 and carbon 4 between two adjacent glucose molecules. How am I going to do? Look, I'll put them together. As always, condensation reaction. How do you do condensation reaction? Take off hydrogen, oxygen, and another hydrogen. What am I going to leave here? I'm going to leave just an oxygen. So I'm going to leave an oxygen here in the middle. And that oxygen will help me to do what? Will help me to link up those two molecules with one another. One more time. So how do you do a condensation reaction between two adjacent glucose molecules? It's like this. Bring the sugars together, close enough, remove a water molecule between the two of them, like this, take off the water, leave an oxygen, and you end up with this bond. So initially I had 2 monosaccharides. Now what do I have now here? I have a disaccharide. What have I done here? I've done a condensation reaction, okay? Good. So again, this bond I've made here, this particular bond I made between sugars, we're going to call it what? We're going to call it 1,4, Y14, based on the carbons. And the name of the bond is going to be called glycosidic. What does glycosidic? Just a name we use for the bonds that you have between monosaccharides. Let's try to do the opposite. Let's say you've been given a disaccharide and you'll be asked to separate them. Okay? So again, here I have 2 disaccharides, and my task this time is to do what? My task is to do hydrolysis. So I already have this glycosidic bond here. I want to break it. How do I break it? Again, my aim is to add water. So I take off the oxygen from one side. Gonna put it here. I put OH and OH. Now they separate. This is what? This is called hydrolysis. Okay. Usually we say 1,4 glycosidic, right? It doesn't make sense. Usually 1 comes before 4 when you count. So I'm not going to call it 4-1. Doesn't make sense. I say 1,4 glycosidic bond. Okay. So what have I done here? I've done hydrolysis. Okay. So let's just show you this one more time. How would you do hydrolysis? Let's say I have disaccharide and I am to break it. I break off the bond and I return back the water. So I've broken this glycosidic bond. Let me just show you what the definition of glycosidic bond. It's a bond you have between two monosaccharides through a condensation reaction. Let's